Pleased to welcome Professor Dr. Jonathan Sweedler from the University of Illinois at Urbana-Champaign. Among his many associations with the campus, Professor Sweedler currently holds the James R. Eisner Family Endowed Chair in Chemistry, is a professor at the Center for Advanced Study and Beckman Institute, and the director of the School of Chemical Sciences. Just recently, he was ranked #1 on the Analytical Scientist Power List, which highlights a tremendous range of talent, ingenuity, and leadership present across all corners of analytical science on a global scale. Professor Sweedler received his Bachelor's of Science degree in chemistry from the University of California at Davis and his Ph.D. from the University of Arizona. Thereafter, he was an NSF postdoctoral fellow at the Stanford University before joining the faculty at the University of Illinois in '91. His research interests are in analytical neurochemistry, both in the development of analytical methods for assaying complex microenvironments and the study of cell-to-cell signaling to uncover novel neurochemical pathways and the characterization of neurotransmitters and neuropeptides from a range of animal models. Professor Sweedler is editor-in-chief of the journal Analytical Chemistry and serves on the Chemical Science Advisory Board. Okay, so let me just stop sharing the screen here. And then, Jonathan, whenever you're ready.
Okay. Hold on one sec, is it? So there we go. I just want to make sure, does it look okay?
It looks great.
Okay. So welcome everyone. In this short talk, I thought I would highlight 3 different vignettes talking about neuropeptides and hormones and some of our work with them. And I decided to pick some work on unpublished work that's all related to some little bit unusual model systems. And so thank you for joining me over lunch, I guess, if you're in the East, and morning if you're in the West. So this is going to be an LCMS-based discovery of peptide hormones in fishes, 2 different species of fish, and picked examples that have to do with the fluidity of sex. So this is, the first example will be Nemo, or clownfish, or anemone fish. This is a fish that can change sex depending on the social organization. So most people have heard, obviously, if Disney had made the movie correctly, if the Nemo's mom had been eaten by another fish, then the dad would become a mom, would change sex. And how does that work? And this is a fairly complex process that obviously depends on social cues and others. And so that is one of the things we've been working on. As we start, I thought I would tell you a little bit, though, background on why these types of measurements are challenging, and a little bit about neuropeptides and hormones. So I use the word, like a lot of us do, neuropeptides, but neuropeptides are hormones or peptides from prohormones that act close, and hormones then circulate through the body, but their biosynthesis is the same. So unlike most proteomics experiments, a neuropeptide is a prohormone made by a gene product, and then it's cleaved by a large range of endopeptidases that are very specific spots. And shown here is a partial sequence of prohormone, and then lysine arginines are cleaved off. Those are the most common sequence motifs that are cleaved, giving you peptides that have these, and then exopeptidase remove those, and you get the peptide hormone. Oftentimes, there's a lot of other steps. If you think about angiotensin now, because of angiotensin converting enzyme, there's a whole host of other enzymes that do extra cleavages. There is a whole bunch of post-translational modifications. Once these are made, they can be released if the hormone's in the soma, or they can actually act as neuropeptides across the synapse, released across a synapse where they actually impact local. And what's interesting is 2 different cells in the same, for example, Oxytocin can be released synaptically and act as almost as a neurotransmitter, even though it's a peptide, a neuropeptide, or it could be released hormonally, and that depends on the specific cell. One of the keys, though, that makes this a little bit different is that oftentimes during this packaging, many of the peptides disappear, and so you don't actually expect to see the entire prohormone. You just see, at times, some of the biologically active peptides. So the first vignette is in collaboration with Justin Rhodes. He's at the Beckman Institute with us, and he works on a large range of models, rats, mice, and others. But one of them is an anemone fish, so clownfish. And looking at the mechanisms that initiate and orchestrate the change from a male to a female. One of the interesting things is that he's even shown that this change occurs first in the brain and then is followed by the gonads. And so it does seem to be, again, a hormonal change. And he enlisted us to try to figure out what those hormones are. And this is just kind of a fun hormones and behavior image that he had in one of his recent articles. So revisiting this, here we have the clownfish, the anemone fish. And how does the brain orchestrate this transformation in response to a social group? Neuropeptide signaling, and I'll show you in the second vignette, is responsible for aggressive behavior, a lot of other plastic changes. And because of other studies as well as the hormones involved, it really is expected that this would involve the HPA, or the hypothalamo-pituitary axis, and the peptides that are present. And so the purpose of our study is first to determine what peptides are present in a clownfish, and then which ones change as it goes to the immature female to the male back to the dominant female. Now, one thing that makes this hard is that when we started this, there was no genome, but even when the genome is first released, there's a lot more mistakes than you're used to thinking about in a mammalian, and it's very poorly annotated. You can't just, for example, search for prohormones or neuropeptides or hormones. And so the first part of this, we work with Sandra Rodriguez-Zas and Bruce Southey. And here are two papers that involved in this. One of them is sort of just a generic approach that we use for model systems where there is no genome or there's a poorly annotated. So we basically, obviously you can do a generalized search, a text search. You can look for proteins and genomic data in related species, in this case, other teleost fish, and I'll show you that in the next slide. And then we can keep on moving through and then doing sequence alignment, phylogeny, and others. And eventually, once we think we have the right prohormone, then we can start to predict what the peptides would be. And that last step is looking at signal peptides and then cleavage sites. And these, this is the dibasic and monobasic sites. And so we've published actually over a decade ago a NeuroPred tool that allows us to make those predictions. That's available on our neuroproteomics website and really can then help refine the search. So let me show you in the next few slides. This is some work that we published actually just earlier this year on the informatics, looking at 22 teleost fish, 11 which related clownfish species, losing quite a few different public repositories. And this allowed us to look for and actually isolate bioinformatically the sequences of about 160 prohormones. One of the interesting things, this is just looking at what we see from the clownfish. Way up at the top, you can see Denio rareo. This is the zebrafish. Only 3 prohormones were found that we can actually find in the clownfish genome. And so really you have to have closely related species. And what's really been fun is the last few years has given us a large number of species. And obviously as you get closer, you would expect a greater number of prohormones. These prohormones then, each one can make 1 or 2 up to dozens of different neuropeptide hormones or neuropeptides that we can then expect to see and measure in the pituitary or the brain. Obviously, now you're thinking, oh, wait, I thought this was a peaks talk or a mass spectrometry talk. We use a lot of different approaches to doing mass spectrometry. This is just showing a few used here for what I'm going to be showing you. We've used the Bruker impact, for example. We've used the different orbitrop systems, the Tribid, and others shown here. Oftentimes, What we do is if we have a sample limited system, we do multi-mass spectrometry because it doesn't require much sample. And so you can spot a fraction of a microliter on a target, make sure the peptides are present when you isolated them, and then we can move on to then doing the LCMS. So I'm not going to go into much detail on this particular aspect of this, but the pipeline is shown here. If you want more information, we had a chapter on this just published a couple years ago in one of the Neuromethod series on neuropeptidomics, in this case, mostly on mammalian, but it's the same system. One thing to think about is the goal of a neuropeptide or a hormone is to bind to a receptor and then be deactivated. And so what that means is they're not very stable. There's a lot of enzymes designed to destroy them. And so if you aren't very careful, you won't see many endogenous peptides because they're degraded. And so what we find is that the first step that we have to do after isolating is to stabilize them. And to stabilize them means to deactivate endogenous peptidases. And so we do that with a detonator or some other heat stabilization approach. And so the quality, it doesn't matter what informatics tool you use, it doesn't matter what mass spectrometry platform you use, if your peptides are destroyed in these first step, then there's nothing you can do to recover them. And so just, it's a single line, but this actually oftentimes determines the success of the measurement. And then we're ready to go. Once we've done this, we now are ready to do either database search with our tailored database or de novo sequencing. From the clownfish, the anemone fish, we from the pituitaries, we were able to identify about 400 peptides from 20 prohormones, 19 prohormones as it says here. And not surprisingly, some of the POMC prohormones, we see the largest number of peptides. It's a very complex prohormone, very well conserved across the vertebrates. One thing you may or may not be aware of, there's 2 POMC prohormones. Teleos fish had a genome duplication, and some of those duplicate genes have been lost and some have not. And so there's actually more complexity in some of the prohormones in some of the fish because of this genome duplication event. And last thing I want to say about this study is that like all other models, if you're talking about mice, rats, humans, or fish, there's a large number of post-translational modifications that are found in neuropeptides. And many of these are critical for either receptor binding or lifetime in after release. And so amidation, for example, or acetylation protect the NRC terminal, and this helps preserve the lifetime. And then some like disulfide bridges and pyroglue and others, and actually acetylation can also drastically change receptor binding. At this point, what we've been able to do is come up with a fairly complete peptide library for the clownfish pituitary. And now what we're just doing is starting a different study now where we're looking at individual or just going to be working with relatively few pituitary, a male and female, to look for peptide differences depending on sex. In this particular case, these studies probably would have been started a few months ago, but our lab is, not obviously been functioning over the last few months because of COVID. And so then we're really excited to be getting these samples from the Rhodes group and continuing this. I wanted to give another vignette just showing you, if you're working with more common, I guess is the wrong term, but a model that has a genome, then in that particular case, you can do things a little bit differently. This is some work with the African cichlid. This is a fish that has males that have 2 different social roles. They have a very complex social behavior. They have territorial males that are colorful and will defend a territory, and then they have non-territorial males that are fairly drab looking. So you can actually see their territorial state very quickly just by looking at the colors. If you remove a territorial male, one of the non-territorial males will quickly, as in almost instantly, display colors and resume and take over that position. And so it's an amazingly fast change in terms of social organization. And then it seems that the brain catches up and changes peptides. And so we were working with one of the experts in this at Stanford University, Russ Fernalda, and we published this work a couple years ago. But this is sort of a complement to the work of the clownfish in the sense that we had sorry about that. We had the genome, and so, but it wasn't, again, as well annotated as maybe what you're used to. So we're able to go through and look at the express sequence tags and use this with the database to come up with a predicted prohormone database from these animals. We actually then looked at annotating these based on signal peptides, cleavage sites, and validated these. And then based on this, we came up with a custom database of prohormones and the major processing enzymes to look for the bioactive peptides. And at that stage, we did the pituitary shotgun peptidomics, as well as de novo. And this allowed us to get a very highly curated neuropeptide database from the African cichlid. And in this particular case, again, even from something as small as a pituitary, we were able to detect over 600 different peptides, and many times many of the expected as well as unexpected post-translational modifications. And in some cases, we did detect peptides with single amino acid substitutions, and this may be due to the fact that we have a population of this particular fish compared to the genome library. And this then is allowing us to then look at the, again, prohormones from this fish pituitary and relate this to behavior. And this is just, again, showing a little bit of the MS conformation of the in silico predicted prohormones. And I apologize if, it's CCK, oxytocin, vasointestinal polypeptide, a lot of the players you would be used to in mammals. Neuropeptides are amazingly well conserved, arginine, vasopressin, and others, and some of these have slightly different names. And so these are the major players then, and this type of information allows people trying to, for example, our collaborators linking social behavior then to neuropeptide hormones, and this will keep them busy for quite a while. So let me end with the last vignette, and this one more is analytical in nature. And this time I'm going to go to my favorite model of Pelagia californica, so not a fish, but a sea slug. Actually, if you were to look at neuropeptide, you would find neuropeptide Y and a lot of other neuropeptides that would be similar. But we wanted to compare how well different search engines work. It's a little scary to say that in a, oops, boy, I'm sorry about this, in a talk here, but I'm obviously doing this because PEAKS works quite well. We looked at it from the peptidomics point of view, and we looked at the false discovery rate in terms of one particular post-translational modification. So if you're used to shotgun proteomics, obviously you enzyme digest if you're not doing top-down, and if you detect more peptides from particular prohormones, it's higher evidence that protein is present. Neuropeptides, you're interested in the individual peptides. You're not digesting. The creature, the cell is digesting with these, and you're interested, with the endogenous peptidases, and then you're interested in individual peptide forms because that determines bioactivity. Minor differences in peptide form can actually change the receptor it binds to. And then the length of bioactive peptides can be 3 or 4 amino acids up to something like insulin, and so up to 50 plus amino acids. And detecting things with 3, 4, 5, 6 amino acids can be quite hard. And so the peptide length has a wide divergence. These are the search engines we did. They use actually different unique algorithms to address the same problem. And so, and in this particular case, we used the same data set. And again, very similar workflow. We took the abdominal ganglia from a Pelagia californica, we desalted. In this particular case, we stabilized, we desalted, we did Orbitrap data, and then we did database. And then we compared the levels. This slide sort of highlights how well the different search engines worked. And in this particular case, again, we're using a curated neuropeptide prohormone library. And this top slide is showing the number of peptides we detected. And the number of peptides then, is up to about, several, 500 or so with peaks, and then this is the number of secreted proteins or prohormones that have signal peptides. And so obviously we get lots of peptides per prohormone. If you wanted to see this depending on size, in terms of peptide length and number of peptides, you can see these violin type plots, or these plots here showing, and in all cases, we did this 3 times, and you can see the FDR cutoff is 1%. We did the same thing when in this particular case with a rat hypothalami, you get very similar results, and you can look at the number of proteins or the number of neuropeptide prohormones or proteins detected in this particular case. So in all cases, one of the reasons, I'm talking here is that peaks in its various forms we now have works exceedingly well for this application. I wanted to end with one interesting observation we've made, and this is about peptide amidation. Amidated peptides, amidation is something that's specific to the secretory pathway. That is, the enzyme that can amidate is PAM, peptidyl amidate monooxygenase, or its 2 subunits, PHM and PAL. In some creatures like C. elegans, it's separate enzyme PHM and PAL. In most vertebrates, it's combined enzyme PAM. This enzyme is an absolute requirement. If you, Betty Eiper, for example, a collaborator, has studied this for 30 years, and if there's not PAM or the PHM PAL or some combination, amidation doesn't occur. If you knock it out, it's fatal, but she's done a lot of conditional knockouts. And this enzyme requires a C-terminal glycine residue to be amidated, and it removes the glycine, and so it leaves the amide. So if you think about this, if it removes the glycine, when you see the peptide, the next amino acid in the prohormone had to be a glycine. If it's not a glycine, it couldn't have been amidated. So this is no different than, say, post-translational modification like phosphorylation that can only occur on things like serine or tyrosine. However, because the requirement is for an amino acid that's not on the peptide, but the next amino acid, this isn't used in most search engines, including peaks. So you can actually ask a question. If you see an amidated peptide and you have the genome, is the next amino acid a glycine? If it's not a glycine, it's a false amidation. As I say, there's a lot of evidence in models ranging from sea slugs, worms, fruit flies, and throughout mammals, humans included, that this is true. We've actually separated amidated peptides with LC into different fractions. Sometimes they're in the same. And you don't see amidation caused by ionization. You don't see this in the non-amidated stays non-amidated, the amidated. This is not a modification that occurs in the gas phase unless, as Scott McCluskey or others have done, they add lots of gas phase basic material or something, and so you can look at this. And so even at a 1% FDR, we actually get a significant number of peptides that are falsely amidated. And so showing you here, the X tandem, for example, got very few hits of amidated peptides, so you don't see many false amidations. So that's an interesting thing. But even when the search engine says it's a 1% FDR, we do see that from a biological point of view, these don't make sense. And then if you go back and look at them, here we have 2 tandem MS, very high quality, and one with poorer sequence, and the poorer sequence one are more likely to come up as falsely amidated. In all cases, though, the spectra did pass the FDR cutoff of 1%. And yet, if you would think if it was a 1% FDR, your false rate would be on the order of 1% and not 30%. And so this is an interesting. Now, obviously, an amidated peptide has a mass change of 1. It's not a very large change, but this is something that I really would hope that search engines would include, the fact that if you have a genome, at least, you can include the fact that the next amino acid should be a glycine. And so the conclusions in terms of this third vignette is that peaks really worked well across the different size. We're excited because it works best with the shorter peptides, and these are actually harder to identify. and had the highest number of identifications. And Peaks also did well with misclassification with amidations, but it still had a higher rate than we would like, given the FDR. And so in the case of something like amidation, you really do have to manually look at the spectra to decide if the assignment fits. Now, I've been talking for about my 25 minutes. I want to end by saying this work was done by, I think I see her on, Dr. Elena Romanova, who has done almost all the mass spec work on the clownfish and the cichlid work. Obviously, a lot of work with Justin Rhodes, our collaborator, Sandra Rodriguez-Zas, and Bruce Southey. I showed a little bit of the amidation work we've worked with and published papers with Elizabeth Eiper on that, and then I showed some of the work with the cichlid fish with Russ Fernald. The last work I showed is some work of a graduate student in my group, Ed De La Toba, as well as Krishna and Apindi. Krishna actually, even though I said graduate student, graduated this May. And so now I have to say Dr. Krishna. And they've done a fantastic job with the work I've shown. And with that, I thank you very much for your attention. And I also should thank the funding agents. And thank you for spending some time with me over lunch or morning, depending on where you're from.